Transient embryonic zones. The formation of the adult cortex is the end product of a series of morphogenetic steps that are initiated in the proliferative epithelium at the surface of the lateral cerebral ventricles. Initial cellular events such as proliferation, migration, aggregation, and selective death of some of the generated cells, as well as the subsequent outgrowth of axons and dendrites, and the establishment of neuronal connections proceed in an orderly fashion in each species according to a species-specific timetable that is regulated by differential gene expression. During embryonic and fetal stages, the telencephalic wall consists of several cellular layers that do not exist in the mature brain. These layers or zones were recognized by a committee appointed by the American Association of Anatomists, Boulder Committee 1970, based on the data from the dissertation on human brain development by the senior author of the present chapter, Rakesh. The committee recommendations for the names of the transient embryonic cellular compartments, termed zones, have been adopted as a generic description for fundamental developmental events for the entire vertebrate central nervous system. However, in the past 4 decades, the development of new techniques, particularly genetic tools for fate mapping and gain and loss of function of targeted genes, have contributed largely to elucidate the genetic regulation of the developmental processes in various species. In particular, the focus has been set on the patterns of expression of transcription factors that seem to exquisitely influence regional differentiation and regulate broad aspects of mitotic activity, fate choice, migration, and differentiation. Recent studies have revealed new types of transient neurons and proliferative cells outside the classical neuroepithelium, new routes of cellular migration, and additional cellular compartments. As a consequence, a revision of the Boulder Committee model that incorporates the new knowledge has recently been proposed. Recently being 2008. In Figure 1.1, the summary diagram from the original Boulder model is reproduced, and at the bottom, the new drawing that incorporates new cell types and cellular zones is shown. Although most of the transient embryonic zones defined by the Boulder Committee were described in the classical literature, for example, Hiss 1904, the subplate zone, SP, has been recognized as a separate entity only relatively recently, 1974. This zone consists of early generated neurons scattered among numerous axons, dendrites, glial fibers, and migrating neurons. Although it has been suggested that the subplate zone provides an opportunity for interactions between incoming afferent fibers and early generated neurons, the significance of these transient contacts is not fully understood. Another suggestion was that the subplate zone serves as a cellular substrate for competition among the initial contingent of cortical afferents, and that this competition serves to regulate their distribution to appropriate regions of the overlying cortical plate. Subsequent autoradiographic, electron microscopic, and histochemical studies revealed that the axons observed in the subplate zone originate sequentially from the brainstem, basal forebrain, thalamus, and the ipsit and contralateral cerebral hemispheres. More recently, the subplate has been shown to be important for the formation of functional architecture in cortex, such as ocular dominance columns in the visual cortex, and also for ensuring the proper formation and strengthening of synapses in this area. After a variable and partially overlapping period, these diverse fiber systems enter the cortical plate. The subplate zone disappears, and most of these subplate neurons eventually degenerate, leaving only a vestige of cells scattered throughout the subcortical white matter, which are known as interstitial neurons. A comparison among various species indicates that the size and role of this transient zone increases during mammalian evolution and culminates in parallel with the developing of association areas of the human fetal cortex concomitantly with the enlargement of cortical fiber systems. The regional differences in the size, pattern, and resolution of the subplate correlate also with the pattern and elaboration of cerebral convolutions. Another transient layer that forms between the neuroepithelium and the peel surface of the dorsal telencephalon that was not mentioned in the Boulder Committee is the preplate, that has been widely used and is included in the new schema. Finally, cell divisions outside the classical proliferative zones have been included.